Hi everyone, this video is about the ABO Blood Group System as well as the other major related blood group systems. It is important for us to determine the blood group of the patient for us to prevent hemolytic transfusion reactions or hemolytic disease of the newborn. Most of the significant antibodies causing these conditions are of the warm reactive type, whereas the insignificant antibodies that we usually detect in the laboratory are of the cold reactive type, meaning they react below 37 degrees Celsius. The warm antibodies are most often IgG, while the cold reactive antibodies are usually IgM. The IgM antibodies are usually of the naturally occurring type, meaning no transfusion or pregnancy is required for their formation. The ABO blood group system was discovered in 1901 by Karl Landsteiner. He and his 5 co-workers began mixing each other's RBCs and serum together and inadvertently performed the first forward and reverse ABO groupings. ABO compatibility is important because ABO compatibility between the donor and the patient is the essential foundation of pre-transfusion testing, and it is the only system with expected antibodies. Whether IgG or IgM, the ABO antibodies can activate complement so they can cause hemolytic transfusion reactions. For HDN, on the other hand, it would be the IgG type of ABO antibodies which are important. Let us now discuss the ABO antigens. The formation of ABO antigens relies on genes at 3 separate loci which control the occurrence and location of the ABO antigens. The presence or absence of the ABH antigens is controlled by the H and the ABO genes. The presence or absence of the ABH antigens on the RBC membrane is controlled by the H gene, whereas the presence or absence of the ABH antigens in secretions is indirectly controlled by the SE gene. So below, you have here the various alleles for each of these genes and the possible combinations. For the H gene found on chromosome 19, you could either have the capital H or the small h allele. Small h is an amorph, meaning it does not code for any protein. So you have here below the 3 possible combinations. You could be homozygous for the capital H allele, or you could be heterozygous, meaning you have one capital H and one small h. Then you could also be homozygous for the small h or the small h allele. For the ABO genes, you could either have A, B, or O as the alleles. And you have here the various combinations. For the secretory gene, you could either have capital C or small c. And again, take note, the small c is an amorph, meaning it does not code for any protein. The antigen, the ABOH and Lewis antigens came from corresponding genes coding for carbohydrate or sugar transferases, which add carbohydrates in a stepwise manner to a backbone protein or lipid. The FUT1 gene makes fucosyl transferase for RBCs. The FUT2 or secretory gene, on the other hand, codes for H antigens in secretions. 80% of individuals have this capital C gene, so they're considered to be of the secretory type. The secretory types again regulated by the capital C or small c genes from an unknown chromosome. And this is important. Okay, the C gene, capital C gene, is also important for the formation of Lewis antigens, later absorbed into the RBC. Later, we'll be discussing about your Lewis antigens. Now, let us discuss your type 1 and type 2 chains. Your type 1 chains are important for the glycoproteins found in secretions and plasma. Type 2 chains, on the other hand, are the precursor of your glycosphingolipids on RBCs. The precursor substance is formed on an oligosaccharide chain, which is the basic structure. So you have here the structure of the A antigen, the H antigen or the O antigen, as described here in the picture, and then the B antigen. So as you could see, both A and B antigens are actually derived from the H antigen. For us to form the A antigen, we need to produce your N-acetyl galactosamine transferase or GalNAc transferase. And the formation of this enzyme, which we also know as the A enzyme, is dependent on your inheritance of the A gene. Galactose transferase, on the other hand, or the B enzyme, is important for us to convert the H antigen to the B antigen. So without the inheritance of an A gene or a B gene, the H antigen remains as is because the enzymes needed to produce the A antigen and the B antigen respectively are not available. Now, some patients, okay, some patients are unable to form the capital H antigen, okay, because they do not have the capital H gene, okay. Their genotype is homozygous for the small h gene. So these are actually what we call the Bombay phenotype. This was discovered by Bende in 1952 and originally found in Bombay, thus the name Bombay phenotype. It is very rare, and this is due to the inheritance of a recessive gene at the capital H locus. Okay? Again, for you to be a Bombay phenotype, you need to inherit two small h genes. You need to be homozygous for the small h gene. Mutation at position 316 from a tyrosine to a stop codon can also produce the O small h phenotype. We also have what we call the parabombase, which is an intermediate form of the capital H gene. The parabombay phenotype is similar to the Bombay phenotype, but the patients have a capital C allele to partially compensate for the lack of capital H. So the phenotypes could be A small h, B small h, or ABA small h. RBCs may type like Bombays, but serum testing shows the presence of your H and A or B antigen, unless they are of the group O type. They may also have the anti-H in serum. The relationship of your A and B antigen is reciprocal, okay, to the quantity of your H antigen, meaning more A or B, if more A or B is made, less of the H antigen remains. The relative amounts of your H antigen differs by the blood group. Among the various ABO blood groups, those of the type O have the most H antigen, since the H antigen is not converted to A or B. This is followed by those of the A2 type, then the B type, then your type A2B, type A1, then type A1B. Type A1B has the least amount of remaining H antigens, since most of the H antigens are converted already to the A or B antigen. The ABO Blood Group System is designated as ISBT number 001. And this is considered to be the most important blood group system. The genes controlling for your ABO system are found on chromosome 9. Okay? The antigens of the ABO blood group are your A, B, and O, or A, B, and H. The antibodies are described to be clinically significant and naturally occurring. The ABO antigen is not restricted to the RBC membrane. The ABO antigens are actually found in all body fluids except the spinal fluid if the individual has inherited a secretor gene. You will also find the ABO antigens in most epithelial and endothelial cells, and even lymphocytes and platelets could absorb the antigens from plasma. The ABO antibodies follow the Landsteiner rule, which is whatever antigens you lack, you automatically have the antibody against this antigen. They are again described to be naturally occurring. They are of the IgM type most of the time, but they could also be IgG or IgA. Newborns do not have antibodies of their own. They start producing the antibodies at 3 to 6 months. This is the reason why we do not perform the reverse typing for patients who are of the very young age, okay, particularly of the neonates. Since in reverse typing, what we are trying to look for are the antibodies. And again, your neonates do not form the antibodies at, until they are at 3 to 6 months old. They can also be of the IgG type. And the IgG type of your ABO antibodies are usually implicated in your ABO-HDN. We also have here a slide showing the various lectins. Lectins are derived from various plants, and they mimic the activity of some ABO antibodies. Okay, like for example, the lectins from Ulex europaeus have anti-H activity. Lectin from Dolichos biflorus has anti-A1 activity. Okay, you need to memorize this lectins since they are being asked in the board exams. We also have here lectins with anti-M activity derived from Iberis amara, anti-N from Visea graminea, okay, and so on. Now let us discuss the various ABO types individually. Those of the group O type, okay, have H antigen. Okay. Generally, this is the most common blood group, and for you to be of the group O type, your genotype should be homozygous for O. You should not inherit A or B. The antibodies of your group O patients could be anti-A, anti-B, and anti-AB. The anti-AB is usually of IgG nature. Characteristically, they react very strongly, and as mentioned, mostly they are IgG. So they may cross the placenta to cause mild hemolytic disease of the fetus and the newborn. This is actually the most common type of HDN. And the second most common type would be your RH HDN. Now, for us to confirm that the patient only has the H antigen, we could use the lectin of Ulex europaeus, which agglutinates cells with abundant H antigen. Group A patients could either have homozygous A genotype, or they could be heterozygous for A. The antigens are A and H, and the antibody that they produce is your anti-B, which is primarily IgM. For your blood group A or blood type A, you could also have subgroups. Okay? For A, you could have A1 or A2 as subgroups. A1 is more common, comprising 80% of the population. A1 RBCs have 4 times more A antigen than A2 cells. Qualitative differences also exist in interactions and shapes of the antigen. Aside from the number of the antigens produced, the antigens of those in the A1 and A2 subgroups actually differ when it comes to structure. Now, this is important for us to know since a small percentage of A2 patients form anti-A1. Okay? So again, a small percentage of A2 patients form anti-A1. This is usually clinically insignificant, but it can cause discrepancies in ABO testing. The lectin of Dolichos biflorus agglutinates A1 RBCs, and this is used to differentiate A1 from A2. Now, this table shows you the differences between A1 and A2. A1 will react with anti-A and anti-A1. Since in A1 patients, you both have the A and A1 antigen. A2 patients, on the other hand, react only with anti-A, okay, because they only have the A antigen. They do not react with anti-A1 since the A2 patients lack the A1 antigen. So that's the basic difference between A1 and A2. For the ABO antibodies, both A1 and A2 subgroups have anti-B, but again, as mentioned, your A2 patients could form anti-A1. Then as for the number of antigen sites present per RBC, as you could see here, A1 patients have more antigen sites, approximately 4 times more compared to A2 subgroups. Then you also have other A subgroups like your A intermediate, A3, AX, AM, AN, AL, and A band 2. Okay, we will not be discussing these individual subgroups in this discussion. For group B, the genotype would either be homozygous for B or heterozygous. The antigens present could be B or H, and the antibody produced is anti-A, which is also primarily IgM. The B subgroups are unimportant and less frequent. So again, they're out of the scope of this discussion. ABO subgroups are usually of weakened or variant expressions of A and or B, and this is attributed to the inheritance of variant forms of transferases. Individuals that have neither transferases, which are inheritors of two amorph genes, are of your group O. To date, 14A alleles, 14B alleles, and 8O alleles have been identified at a molecular level. Lastly, you have your group AB, which is the least frequent, okay, comprising around 4% only of the population. The antigens present are A and B, and you could have some H antigen also. This could be further subdivided into A1B or A2B depending on the status of the A antigen. As for the antibodies, they do not produce ABO antibodies. Now for ABO testing, there are two types. You have your cell typing or serum typing. Cell typing is known as forward grouping, wherein the patient RBCs are agglutinated by known sera. So for forward grouping, what we are trying to detect are the antigens present on the RBC of the patient or of the recipient. For serum typing or reverse grouping or back typing, the patient's serum or plasma, okay, is added to A1 and B RBCs. So what we are trying to look for here are the antibodies of the patient, not the antigen. This is the reason why, again, we do not perform reverse typing on neonates because neonates lack the antibodies. Now, you have to note that for forward and reverse typing, there are opposite reactions. If the forward reactions are not opposite of the reverse, then you have an ABO discrepancy. Both serum and cell typing are required unless testing babies less than 4 months of age or confirming testing done elsewhere. So this is a very important table showing you the expected results for your forward and reverse typing. Okay, so for group A patients, you have a reaction with anti-A since you have the A antigen. You would not be reacting against anti-B since you do not have the B antigen. With the addition of A1 cells to the plasma of type A patients, there would be no reaction since you do not have anti-A. What you have is anti-B, which reacts with the B cells, thus the reaction, okay, under your B cells. For your B group, okay, for your type B, the reaction is found in anti-B since you have the B antigen. Then, in reverse typing, you have a reaction against A1 cells, okay, since the type B patients produce anti-A1. For AB patients, both anti-A and anti-B react. Okay. For reverse typing, you do not have reaction since your type AB patients do not produce ABO antibodies. For group O, forward typing produces no reaction or no agglutination. For reverse typing, both addition of A1 and B cells produce agglutination since both anti-A1 and anti-B are present. Okay, so this is just another way of presenting it. ABO discrepancies are detected again when you have a discrepancy between the reactions or between the results of your forward and reverse typing. Okay, so there's this agreement between the interpretations of forward and reverse grouping. These are generally caused by either antigen or antibody problems. So on your notes, you have their comprehensive list of the various ABO discrepancy types, as well as the examples of conditions causing these discrepancies. Please study the table or study your list on your notes. Acquired B phenotype, okay, is seen in colon cancer, intestinal obstruction, and gram-negative sepsis. In forward typing, the patient is seen as AB, type AB, but in reverse, they are reacting as type A. The reason why patients acquire the this acquired B phenotype is because of bacteria which deacetylate group A sugar, okay, converting the group A sugar into a group B sugar. Okay, and this is usually transient. Once the condition is removed, once the condition of the patient has improved, we will see that this discrepancy eventually disappears. Okay, so this is an example of a patient with acquired B phenotype. So in forward grouping, as you could see, the interpretation is AB, but for reverse grouping, it's type A. Now, consequences of ABO incompatibility. The main reason, again, why we try to determine the ABO type of the patient is that we're trying to prevent HTRs and HDN. Severe acute HTRs are the most frequent blood bank fatalities, and they're usually because of clerical errors. ABO HDN is also the most frequent HDN type. However, compared to the RH HDN type, this is usually mild. Okay? So ABO HDN happens if the mother produces anti-AB, which is usually IgG. And recall that anti-AB is only produced by those of type O. So ABO HDN happens among type O mothers. This anti-AB IgG crosses the placenta, then it binds to fetal RBCs, causing now hyperbilirubinemia. Okay? If you check the cord cells of the baby, you would see that the RBCs, okay, the cord RBCs, cord blood RBCs are DAT positive, meaning they have been the RBCs of the patient have been sensitized already. Okay, the fetal cells are coated with anti-A reactive antibodies. So let us now discuss the major blood groups associated with the ABO system. First is the Lewis blood group system. The Lewis blood group system, okay, is comprised of antigens secondarily adsorbed to RBCs. So they are not found primarily on the RBC membrane. The capital Le gene produces your Lewis A antigen. Then this Lewis A antigen is then converted to Lewis B through enzymes produced by secretors. So this table summarizes the various combinations of your Lewis genes and your secretor gene. So as you could see, if you have the capital Le gene as well as the capital C gene, your phenotype would be Le A negative and Le B positive. Okay? The reason behind this is that if you have your capital Le gene, the Le A antigen is produced, but then it is subsequently converted to your Lewis B antigen. Now, if you have inherited your capital Le, but you are a non-secretor, the Lewis A antigen remains as is. So your phenotype would be Le A positive, Le B negative. Now, if you are homozygous for your small Le gene, okay, you will not produce your Le A antigen. So you will not be producing Le B also. So your phenotype would be negative for both Le A and Le B. If you inherit a small Le, then a small C, again, you would be negative for both. Or if you inherit Le, but you're a secretor, you will still be negative for both. Okay, again, for you to be able to produce your Le A antigen, you need the capital Le gene. So the newborns are Le A negative, B negative. At 2 weeks to 6 months, if they have capital Le as well as capital C, they would begin producing your Le A gene, or Le A antigen rather. Then, this will transition to A positive and B positive. Then eventually, most or all of your A antigen would be converted to B. So your phenotype would be A negative, B positive. If a person is homozygous for the small gene, they will have no Lewis antigens in plasma or on RBCs. Lewis antigens decrease during pregnancy, so pregnant patients may have transient and significant Lewis antibodies. La-A negative, B positive people don't make anti-La-A. These people still have La-A, just not on their RBCs. Children's Lewis type may vary as antigen chains are converted. So again, the Lewis antigens are your La-A and La-B, and La-B is seen more frequently. Lewis antibodies are described to be naturally occurring. They are primarily found in your A negative, B negative phenotype. Antibodies which could be produced include your anti-La-A, anti-La-B, and anti-La-X. They are described to be cold reacting IgM. They may produce HTRs rarely, but they will not produce HDN since they are of the IgM type. Remember, the IgM does not cross the placenta. As for the frequency, among whites, the most common phenotype is your A negative, B positive. This is also true for blacks. The next blood group system is your I blood group. The antigens of your I blood group are built on chains related to your ABO, and the expression is age dependent. Neonates produce small I antigen. Then as we grow older, we produce more capital I antigen. So remember, big I in big people, little I in little people. Antibodies are cold reacting IgM. They are naturally occurring, and some autoantibodies have specificities for the antigens belonging to this blood group. So classic associations, your auto-anti-capital I is found in cold agglutinin disease, and this usually follows after Mycoplasma pneumoniae infection. Auto-anti-small I, on the other hand, is associated with infectious mononucleosis caused by Epstein-Barr virus, and this is less often a problem compared to your auto-anti-capital I, or anti-big I. The P blood group system is also ABO-related. The antigens are built on ABO-related chains. The most frequent antigen belonging to this blood group system is your P1 antigen. Okay? So this is the most frequent. Other antigens belonging to this blood group system include your P and PK. A combination of these three antigens makes the P phenotype. The most common P phenotype is your P1. So your P1 phenotype is positive for both P1 and P antigen, but negative for the PK antigen. Very rarely, people lack all three antigens. These individuals who lack the three antigens can make anti-P, P1, PK, which is associated with acute HTRs, HDN, and early abortions. The P antigen, please take note, is the parvovirus B19 receptor. Anti-P1 is cold reacting, naturally occurring, and is insignificant. And it could be neutralized by hydatid cyst fluid as well as pigeon egg fluid. There's also association with PCH, okay, or paroxysmal cold hemoglobinuria. Anti-P1 or anti-P rather, okay, can be produced by patients with PCH. This is described to be biphasic, okay, biphasic and IgG in type. Okay, and this is also known as your Donath-Landsteiner biphasic hemolysin. Okay, why is it called biphasic? It is called biphasic because in cold temperature, it binds complement. Then in warm temperature, it activates complement. So there are two separate activities in two different temperature settings. So that's it for this video.